this data set in this lecture and in the which is about doing some classification or about images with traditional methods, and we will use the same data set for the rest of the course, I will say, to do also this and some unsupervised learning. So this is a basic of neuroscience image analysis. Neuroscience, by the way, is a technique. Well, it's not a technique. It's a field of study that has had to deal with big data since the 90s, actually. And there have been published lots and lots of papers that indeed do not resist the scrutiny of time, given the number of false positives that they got. But there are many, many things that do work instead. Now, what's the single basic concept behind the neuroscience? It's the existence of a standard brain. What's the concept of the standard brain is that the same place, whatever that might mean, in the brain of each human does the same task in everyone. And that's a pretty strong assumption, but up to a point it is true. And this is a typical brain. The shapes are very similar between everyone. And you can recognize a couple of features. And like this central sulcus here defines the motor cortex, which is devoted to control the motion. This is, next to it, there is the sensory motor cortex. This is the auditory cortex, the visual cortex. And you can guess from the names that they have been found experimentally over time, each of them to be devoted to some specific task indeed. There is a representation called the homunculus, given its weird shape and its distortion, that takes for the motor cortex and the sensory cortex an association between the place along the cortex and what it's connected to. And you can see how distorted it is, but it makes sense, because if you think about the tactile sensitivity of your back, you don't really feel much, while if you're working with your fingers, you have extreme sensibility. You can produce, if you are a watchmaker, you can deal with microscopic gears without any problems. There's really a lot of neurons devoted to controlling the muscles of the mouth. You are able to speak. You are able to do lots of facial expressions. And on the contrary, for your calf, which can go up and down, you have very little neurons. And so on. These are two representations, and you have, and this has all been found over a very long field of research, not only with functional MRI, but even before that with surgeries, with people that lost some part of the brain to a trauma, but I mean an impact or due to a tumor. It was found that lots of parts of the brain have specific functions. And by the way, then this was taken to the extreme. Now, maybe 15 years ago, there were a lot of neuroscientists saying, where does fear reside in the brain? But the fact that the inputs and outputs of your brain are in some specific locations in bundles of neurons, it does not mean that the processing is located in one part of the brain. Emotions, reasoning, and so on can be complex factors of interaction. They don't need to be localized, and it doesn't necessarily make sense. Some might be, some aren't. But these findings here related to the inputs and outputs are extremely well established. So where do we start from? From books that were sold in the 70s, 80s, and even early 90s. You could buy a book of Brain Atlas, like a physical map, there was the so-called system of Thalayerk brain coordinates. It was based on a single post-mortem studies. The concept was that of relative distances, which work pretty well because if you are a bigger person, you generally have a bigger brain, but the relative differences kind of stay the same. And so you get your pathology slices. The pathology is when post-mortem you extract slice and look at under a microscope. You could, you took the image, you went there with a ruler, an actual ruler, then went to the atlas and said, okay, this is probably the frontal cortex. Nowadays we have something more which has been better described. It's the Montreal Neurological Institute coordinate system. And what's generally used is the MNI 305, which is it starts from 305 scans of normal brains, which have been linearly co-registered, which means rotated and translated, to another 241 brain that had been roughly aligned to this previous Thalayerk brain, because we set a standard and we don't really want to dismiss it. And it was based on T1 images only. Nowadays, if you go to that's actually, I did it 2 or 3 years ago, and you go to the Montreal Neurological Institute website, you get a list of possible template atlases, and they are all very close to each other, but still there are some very minor differences. Some features are 1 or 2 millimeters away in one brain to the other. Now, what you do about that, I will give you a link to a presentation that explains that, given that many of you asked me about this process during previous lab experiences, is that you have your image and you align that to this brain. The brain is the most rigid part of the body. It's within the bones, the brain bones. I don't know the English term. And therefore, in general, if you do, within the same subject, if you do a translation and a rotation, you get perfect alignment, not so much maybe over many years. But then you also need a deformation, a more elastic deformation, and there are thousands of ways to do that because it's not a very well-defined problem, so you can have, of course, 1,000 solutions with minor differences and no true solution. And it's still an optimization problem. You have 2 images, you need to find the similarity metric and optimize that. Now, when using any of these methods, which is an important part, but we will not focus on that. You aligned your brain, your image of one brain to this template reference. You can load atlases, and they are defined by consortia, and they have been painstakingly manually drawn according to some criteria, which can be anatomical. You know, I've shown you the central sulcus, it's pretty easy to recognize. And then you start to define lots and lots of geometrical features. Or functional, with some kind of studies, you find that a certain area does some task, and then you divide according to that task. Or some architectonical markers, there are multiple ways. Often they overlap, but sometimes not. The boundaries are not so easy to define. So this is a very standard visualization software. When you have your image aligned in this reference space, you can overlay this atlas, one atlas of your choice, and by just navigating with your mouse, it will tell you, like here you are in the, okay, here with the pointer, I'm in this red part here, and the software tells me that's the post-central right cortex, if I care about it or not. Why was that super important? Because it allowed the really base of neuroscience analysis, which even now, with artificial intelligence, we still care about, because AI might be effective, but neuroscience very often wants to study the mechanism. So you want mechanicistic explanations. You don't want something that spits out an output. You already have the brain for that. So what was the pipeline for really all kinds of neuroscience analysis? You acquire lots of images of the brain, of all possible tasks. You align them to a standard space. You do some heavy smoothing, so for multiple reasons. And one of that is to reduce noise, and the other one is to not look at individual anatomical small differences, but only at large-scale effects. And then if you are, not only you align, but even reslice them, so your images are aligned pixel by pixel, the matrix is really working on the same pixels, you can do a general linear model statistics, like literally a t-test. You can do the student's t-test, a correlation analysis, or an analysis of variance at the pixel level. You straightforward do that. After that, what you need to do is the really most difficult part. You estimate statistical significance. A brain, I don't know, let's say it's 10, let's say it's a 10 by 10 by 10 centimeter cube. So if you have, it's about 1 liter if you do this approximation. If you divide it in, you get like 1 million pixels if you're working with 1 millimeter pixels, or maybe 100,000 pixels if you're working with 2 millimeter pixels, which makes more sense. And then you have 100,000 students t-test, which, by the way, so you get lots of false positive, for sure. Also, each pixel is, given the smoothing that you performed, each pixel is related to the neighboring ones. So estimating the effective p-value correcting for multiple comparisons was really, really hard. But it can be done. There's a theory that does that. It does an effective number of study. You can also do that within the brain of a single patient. Like if you do functional MRI, you see the amount of oxygen in the blood over time as responding to a stimulus. You get it, you don't register to an atlas, you just do it of the single patient. You have 1 million images over time, and you do the t-test between images during the stimulus and images without the stimulus. And now we will do that with PET images, but you can also, it's popular to do that even with volumes of gray matter. You take the image, you extract the gray matter, and then define some kind of localized concentration of gray matter and do a localized t-test. So it's pretty powerful. This is all to introduce the data set that I will provide you. It's a data set about dementia and FDG PET. It turns out that different types of dementia are associated to damages to specific areas of the brain. Also, because the cell's metabolism, the ones that you can study using glucose, is one of the first aspects to be impacted. Let's introduce the Alzheimer's disease cascade hypothesis. This is at histology post-mortem, the comparison of a healthy brain and a severe Alzheimer's disease brain. You can see that it has shrinked massively. But in this representation, that's even now our best assumption about how this works. You have all of these phenomena appearing in order. And this is when you can start to detect that. And you have first some amyloid in the cerebrospinal fluid, which, however, is not super specific than amyloid accumulation in the brain, which you can see with PET, tau protein accumulation in the cerebrospinal fluid. damages seen in FDG. Then only here, which we have experimental indications that this can mean 10 years, 15 years. You have what's called mild cognitive impairment, which is a kind of a very broad condition. You have a person which is not as bright as it used to be. For Alzheimer's disease, this is especially some short-term amnesia. But however, the symptoms are extremely not specific. You have some people that start having some minor loss of mental acuity without ever converting to Alzheimer's. When getting a different dementia, might have a problem due to perfusion of the brain, the amount of blood that they get, and this is all not Alzheimer's disease. But the thing is that when you are sure that it's Alzheimer's disease, most of the brain cells will have already died. So it's clear that if you want a drug for that, you need to act like here and not here, which is a decade before, however, so it's really, really hard. There are many other kinds of dementia. You have one typical, not so frequent other kind of dementia is dementia with Lewy body, which is, it's called alpha-synucleopathy. And again, a problem with lots of dementia is that the symptoms are not specific at all. Here we have neurological and psychiatric symptoms. It starts with mood swings, hallucinations, starting to be unorganized. That can really be everything. Frontotemporal dementia of the behavioral variant is characterized again by unspecific symptoms. However, this generally progresses pretty fast. And it's lack of inhibitions, lack of interest, difficulty in keeping jobs, repetitive behavior, and lack of inhibition can really turn people around in their 70s, getting them some really inappropriate behavior from a church-going lady, which at 70s, while in a retirement home, completely flips around. And if you do that kind of t-test that I was telling you, you will see that when you analyze the PET with FDG, this analog of glucose, you see that you get a very specific spatial sign. Frontotemporal dementia, you get the frontotemporal lobe, as the name says. And this is caused by the tau protein. Lewy body dementia is turns off the occipital, the parietal part of the brain, like AD. The parietal part of the brain is this one. But however, it also involves the occipital lobe quite a lot. And okay, just to recap, that's the pre-processing steps that happen. We have some, you need to spatially normalize the image to this MNI space. When I was talking about spatial smoothing, you filter with actually like 8 millimeter FWHM filter to avoid seeing local effects and reduce the number of multiple comparisons and so on. I've glossed over that, but you need to scale the intensities. You have a PET image which is in some kind of random intensity, and you need to scale that. You can either do that with the global mean of everything that's in the brain using a reference region, or you could see in the notebook that I've given you another suggestion also. And where do we get the data from? Well, that's actually I will give you different data, but I want to introduce this project, which is an amazing project. It's the Alzheimer's Disease Neuroimaging Initiative. So this is the characteristic of this project. Probably you should be recognizing all of these terms from Professor Bernasconi's course. It's a multi-site study, so data are being acquired over lots of hospitals. Longitudinal, it means that you follow each participant over time. Prospective, so you first decide how to collect the data, then you collect them, not you get the data that you find around and then process them. Naturalistic, so it means there is no intervention whatsoever. Study of normative cognitive aging, mild cognitive impairment, and early Alzheimer's disease. It collects longitudinally MR images, mainly T1 volumetric, but also many other kinds of images, and which ones might change over the years. Cerebrospinal fluid biomarkers, which are very important for dementia. PET images, starting with FDG. Now they also acquired amyloid load and the tau protein. Many blood biomarkers. Neurological tests, psychological assessments. Over time, most of these studies, the ones that don't involve radiation, are required for each person every 6 months, over years. And even very complex demographics, they started acquiring like information about education, having been living with a partner or not, divorcing, ethnicity, and kind of job, the intellectual load of the job, and so on and so on. And to give you an idea, it has been divided in phases. We are in phase 4 now. It's still ongoing, and it started in 2004 by acquiring 200 normal subjects, 200 AD subjects, and 400 MCI subjects. There are some subjects for which you find like 8. These are few, but you find some subjects that have 8 years of follow-up. Both healthy and, well, not Alzheimer, but maybe MCIs that never convert to full Alzheimer. So it's a very precious resource, and you can do some really some kind of big data study that intersect multiple domains. Nowadays, they also acquire genetics and so on. However, dealing with the data can be really hard. You really need to work with pandas a lot. Because even if I give you a classification, I tell you, predict the future of this subject, and it's not that trivial defining who will convert and who not. What do you use a cutoff? Which measure do you use? There are some curated data set out there. There was a challenge some years ago, which was called TEDPOL. Okay, and that's it. And I have... Okay, and I've uploaded here some the data. It's PET images already being registered, aligned voxel-wise, same number of voxels, and so on. If you were a good programmer, you will put some checks maybe when loading, but these are just exercises. And we have 61 AD subjects. The truth is in the label of the directory. 65 behavioral variant frontotemporal dementia subjects, 58 Lewy body dementia subjects, 112 healthy controls, and some masks of specific regions of the brain that you might use as a reference region. We have a mask of only the neurological structure, because here I give you a rectangle. The pons, which is the stem of the brain, the supramotor cortex, the cerebellum, some white matter. And then we have the notebook. I don't have it open here. Let me open it. Classify AD. And so today, for the hour and something that is left, I will ask you to play around with some traditional analysis. I will ask you to try to really run the voxel-wise t-test. It will take like 20 lines of code. It's relatively easy, and so you can see how it works. And then if you want, you can do task 3. You extract some standardized patterns and see how much each subject expresses that and how you can, well, you can classify. And more interesting, somebody asked me for that in previous lectures, you can also do some direct strategies. The brain neuroscience is like the only field in which it works, given that you can realign images. But it works surprisingly well. So what you could do is take the data and do a PCA. See what happens with that. You will probably need to do some intensity scaling, or you can play with doing a log transform beforehand, but it will be really interesting to try that. Okay, any questions about this task? Then in the next lectures, we will try to apply some AI-based classification, especially unsupervised.